started. We're covering two chapters tonight, chapters 13 and 14. So we're going to dive into evolution tonight. Let me share my screen. We will get started. Okay, so we're going to first look at populations and how populations evolve. So let's talk about the diversity of life. For all of human history, people have named, described, and classified the inhabitants of the natural world. Well, there's a branch of biology called taxonomy, and it's concerned with naming, identifying, and classifying species. Now, the Linnaean system includes a method of naming species and a hierarchical classification of species into broader, larger, or broader groups of organisms. So Linnaeus has also introduced a system for grouping species in categories. And so in this Linnaean system, each species is given a two-part Latinized name, or we call it a binomial. The first part of the binomial is the genus, which is a group of closely related species. And then the second part of the binomial is used to distinguish the species within a genus. So grouping organisms into broader categories is a way to structure our understanding of the world. So in this example, you can see for this Panthera, starting at the very big, large domain, it would be Eukarya. And as you go up, you go to the more specific species, which is the Panthera. So how do you explain the diversity of life? The explanation of the origin of diversity of life was accepted by present-day biologists in the evolutionary theory that was proposed by the scientist by the name of Charles Darwin. And he wrote a book on the origin of species by means of natural selection, published in 1859. Now, there was also this idea of fixed species, and the Greek philosopher by the name of Aristotle generally held the view that species are fixed, permanent forms and do not change over time. It aligned with the Judeo-Christian culture, which reinforced the idea of the literal interpretation of the biblical book of Genesis, which tells the story of each form of life being individually created into its present day form. And then, of course, you know, with Noah's Ark, each species was actually placed on the ark to survive the flood, right? So that was his idea. All right, so in the 1600s, religious scholars estimated the Earth's age to be about 6,000 years. And the idea that all living species came into being relatively recently and are unchanged in form dominated the intellectual climate of the Western world for centuries. At the same time, though, naturalists were grappling with the interpretation of fossils. And fossils are the imprint or remains of organisms that lived in the past. So basically, fossils perplexed those naturalists in the 1800s because you had these snake stone or a skull, and they don't look like anything in present day form. They found fossils and sedimentary fossils that are formed by minerals replacing the organic matter of a tree. This is a 45-million-year-old insect that it was embedded in amber. You have excavation of skeletons. You have actually the organism that has been discovered, and then of course imprints. So evidence from fossils. Not all fossils are the actual remnants of organisms. Fossils can be cast, formed when a dead or buried organism decomposes and leaves an empty mold that is later filled by mineral dissolved in water. It can be an imprint, such as a footprint or burrow that remains after the organism decays, or it could be coprolites, which are fossilized species. So paleontologists are scientists who study fossils. And the fossil record is the ordered sequence of fossils as they appear in the rock layers, marking the passage of the geologic time. So the younger strata is on the top, and then of course, the older layers, the relatively age of the fossils can be determined by the layer in which they're found. So as Darwin acknowledged, the fossil record is incomplete. And he said it's incomplete because of the following. He said that many Earth's organisms did not live in areas that favored fossilization. Many fossils did not form that did form were in rocks later distorted or destroyed by geological processes. And not all fossils that have been preserved are accessible to paleontologists. So you have this other scientist that came on. This naturalist compared fossil forms with living species and noted patterns of similarities and differences. So in the 1800s, Jean-Baptiste de Lamarck suggested that life evolves. So he explained evolution as the refinement of traits that equip organisms to produce to perform successfully in their environment. So he proposed that by using or not using body parts, an individual may develop certain traits that pass on to his offspring. All right, so let's talk about Charles Darwin. Well, he was born more than 200 years ago on the same day as Abraham Lincoln. He was fascinated by nature. He quit medical school. He studied clergyman at Cambridge University, and then he began a sea voyage at the age of 22 on the HMS Beagle and helped frame the theory of evolution. So the Beagle was a survey ship that charted poorly around the stretches of South American coast. He spent a lot of time exploring the natural world, collecting thousands of specimens of fossils and living plants and animals. He kept very detailed journal of his observations and noted carefully noting the characteristics of plants, animals, and what made them well suited in these very diverse environments. So this is his voyage. So what did Darwin observe? Well, many of Darwin's observations indicated that geographic proximity is a better predictor of relationship among organisms than similarity of the environment. So what does that mean? For example, the organisms living in temperate regions of South America more closely resemble the species living in tropical regions of that continent than species in temperate regions of Europe. So he also found, Darwin found that though clearly examples of species different from living ones were distinctly South American in the resemblance to the contemporary plants and animals of that continent. So he went on and was very intrigued by the geographic distribution of these organisms on the Galapagos Islands. Most of the animals that inhabited these remote islands were found nowhere else in the world, but they did resemble South American species. One example are the Galapagos marine iguanas. They had flat tails that aided them in swimming and were similar to, but not distinct from, the land-dwelling iguanas on the islands and on the South American mainland. He also noticed that each island had their own very distinct variety of giant tortoises. So he gained new insight. went back to England and reflected on his observations and analyzed his specimen collections, discussed his work with colleagues, and concluded that the evidence was better explained by a hypothesis that present-day species are the descendants of ancient ancestors, which they still resemble in some ways. So over time, these differences gradually accumulated by a process that Darwin called descent with modification, to phrase his description of evolution. So he proposed a scientific mechanism for how life evolves, and the process is called natural selection. So in natural selection, individuals with a certain inherited trait are more likely to survive and reproduce than are individuals with other traits. So he hypothesized that as the descendants of a remote ancestor spread into various habitats over millions and millions of years, natural selection resulted in diverse modification, or what he calls evolutionary adaptations, that fit them to specific ways of life in their environment. So he spent the next 2 decades compiling and writing about the evidence of evolution. And then you had some other scientists by the name of Alfred Wallace doing fieldwork in Indonesia that had a conceived hypothesis almost identical to Darwin's. So then he finally published his book on the origin of species, a book that supported his hypothesis with the immaculate logic of hundreds of pages of evidence drawn from observations of experiments in biology, geology, and paleontology. So the hypothesis of evolution set forth the origin of species has been generated predictions that have been tested and verified for more than 150 years of research. So with that, scientists regard Darwin's concept of evolution by means of natural selection as a theory, which means it is widely accepted explanatory idea that is broader in the scope than a hypothesis, generates new hypotheses, and is supported by a large body of evidence. Okay, so let's switch to the evidence of homology. So similarities resulting from common ancestry is known as homology. And so characteristics present in an ancestral organism are altered over time by natural selection, and it is a descendant face as the descendant face different conditions. As a result, related species can have characteristics that are similar, yet function differently. Darwin cited the anatomical similarities among vertebrate forelimbs as an evidence of common ancestry. So let's take a look at these forearms of the human, then look at the cat, look at the whale, and the bat. So again, these are homologous structures and anatomical signs of descendant with modification. So advances of molecular biology provide a much deeper understanding of homology. The evolutionary history of each species is documented in DNA inherited from its ancestral species. So if 2 species have a homologous gene with sequences that match closely, biologists conclude that these sequences must have been inherited from a relatively recent common ancestor. And of course, the greater the number of sequence differences between a species, the more distant their last common ancestor. So molecular biology provides a strong evidence that all life forms are related. And so all forms of life, as we know, use that same language of DNA and RNA. And those genetic codes, how RNA triplets are translated into amino acids, is essentially universal. So organisms as dissimilar as humans and bacteria share homologous genes inherited from a very distant common ancestor. Now, vestigial structures are remnants of features that serve important functions in an organism's ancestor. I want to give me 1 second. Let me make sure. Okay. I want to make sure I was recording. Okay, sorry. Important functions of an organism's ancestor. These include a small pelvis and hind leg bones of ancient whales, eye remnants in blind species of cave fish, and then goosebumps in humans caused by small muscles under the skin that make body hair stand on end. So homology can also explain observations about embryonic development that are otherwise puzzling. For example, at some point in the development, all vertebrate embryos have a posterior tail to the anus. They have structures called pharyngeal or throat pouches that may be gills in fish or parts of the ear in humans. So here, this is an example of the chicken embryo and the human embryo, and how you see that at some point, both embryos have that post-anal tail and pharyngeal pouches. They look very similar. So Darwin visualized the history of life as a tree, and biologists illustrate these patterns of descent with an evolutionary tree, although today they often turn the tree sideways to be read from left to right. So homologous structures, both anatomical and molecular, can be used to determine the branching sequence of an evolutionary tree. And evolutionary trees are hypotheses reflecting our current understanding of patterns of evolutionary descent. So this is an example of an evolutionary tree of a pteropod, which are four-limbed animals. Each branch point represents a common ancestor of the lineage beginning there or to the right of it. And then the blue dot represents a homologous trait shared by all the groups to the right of the mark. So there are some key points about natural selection. Let's talk about those. So first, natural selection affects individual organisms, but individuals do not evolve. A population, which is the group of organisms, evolves over time as an adaptive trait becomes more common in the group, and other traits change or disappear. So with that, evolution refers to generation changes in population. Thomas Malthus contended that human disease, famine, and war were the consequences of human population increasing faster than the resources. So Darwin applied Malthus' idea to populations of plants and animals, reasoning that the resources of any given environment are limited. The production of more individuals than the environment can support will lead to struggle for existence, and only some offspring survive in each generation. So in this process of natural selection, individuals whose traits better enable them to obtain food, escape predators, or tolerate physical conditions will survive and reproduce more successfully, passing these adaptive traits to their offspring. So Darwin reasoned that if artificial selection can bring about significant change in a relatively short period of time, then natural selection could modify species considerably over hundreds or thousands of generations. So when we see natural selection in action, biologists have documented evolutionary change in thousands of scientific studies. And unfortunately, an example is natural selection of natural selection in action is the evolution of pesticide resistance in hundreds of insect species. So typically the pesticide should kill insects, but unfortunately, through evolution of the insect, the chromosome with the allele that allows for that resistance to occur becomes present, and then that insect reproduces, survives and reproduces, and so now you have hundreds of insects that are now pesticide resistant. So natural selection can amplify or diminish only heritable traits, although an organism may, during its lifetime, acquire these characteristics that help it survive. Acquiring characteristics cannot be passed on to the offspring. Natural selection is not goal-oriented, and it does not lead to a perfect adapted organism. A trait that is favorable in one situation may be absolutely useless or even detrimental in different circumstances. And so let's talk about that mutation, that change, okay? We talked about it in our previous unit, but we know that mutation is the ultimate source of genetic variation that serves as raw material for evolution. So new alleles originate by mutation, and that's a change in the nucleotide sequence, right? Nucleotide sequence of DNA. In multicellular organisms, however, only mutations in cells that produce gametes, that's the sperm or the egg, can be passed to the offspring and affect its population's genetic variability. A change as small as one single nucleotide in a protein-coded gene can have significant effects on the phenotype. For example, sickle cell disease, as we talked about in our previous unit. So mutations that affect a protein's function will probably be harmful. Now, on rare occasions, a mutated allele may improve the adaptation of an individual to its environment and enhance its reproductive success. This kind of effect is more likely when the environment is changing in such a way that mutations that were once disadvantageous are favorable under new conditions. All right, so let's talk about sexual reproduction. In organisms that reproduce sexually, most genetic variation in that population results from a unique combination of alleles that each individual inherits. Remember, in humans, for example, you get 23 chromosomes from mom, 23 chromosomes from dad. Well, fresh is assortment of alleles, of existing alleles arise every generation from 3 random components of sexual reproduction. You have independent orientation of homologous chromosomes at metaphase 1 of meiosis. You have crossing over that occurs, and then random fertilization. So the evolutionary impact of natural selection is apparent only in the changes in population of organisms over time. And remember, a population is a group of individuals of the same species that live in the same area and can interbreed. So different populations of the same species may be geographically isolated from each other to such an extent that an exchange of genetic material can never or rarely ever occurs. Such isolation is common in populations combined to different lakes. So on different sides of the lake, you will find that those are isolated populations. And at the population level, biologists focus on the gene pool, which consists of all copies of every type of allele at every locus in all members of the population. For many loci, there are 2 or more alleles in the gene pool. So now, what are mechanisms of evolution through natural selection? What mechanisms can change a gene pool? Well, there are 3 main causes of evolutionary change. We have natural selection, which we talked about. You have genetic drift and gene flow. So natural selection is the most important because it is the process that promotes adaptation. When we talk about sexual selection is a form of natural selection in which the individual with a certain trait are more likely than the other individuals to obtain mates. So you have sexual dimorphism, which basically means that there is a distinction in appearance between males and females, not directly associated with reproduction or survival. In a more common type of sexual selection, the individuals of one sex, which is usually the females, are choosy in selecting their mate. So you find sexual dimorphism where the male and female look distinctively different. And then you also see, in this case, two males competing for territory and for the mates. Now, there are three general outcomes of natural selection. If we, for example, if we were to graph the coat color of a population of mice, we would get a bell-shaped curve. If natural selection favored a certain fur color, phenotypes over others, the population of mice would change over generations, and three general outcomes are possible. So the three possible outcomes, just looking at fur color of the population of mice, is directional selection, where there's a shift of makeup of population selecting a favor of one extreme. Disruptive selection, which can lead to a balance between two or more contrasting phenotype forms in a population. That's the one that you need to remember. And then stabilizing selection, which favors intermediate phenotypes, occurs in relatively stable environments and is the most common. But I just want to bring your attention to number 2 again, which is disruptive selection. And that is letter B, disruptive selection. All right. So we have a rising threat, just like with pesticides, or I should say, insects that are now pesticide resistant. You also have antibiotic resistant bacteria. So antibiotics, as you know, are drugs that kill infectious microorganisms. And in 1940s, penicillin was the first widely used antibiotic. But shortly thereafter, in 1952, not long, some bacteria had genetic traits that enabled them to resist the killing power of drugs. So in the same way, pesticides select for resistant insects, antibiotics select for resistant bacteria. So our enthusiasm for curative powers of antibiotics has encouraged the evolution of antibiotic-resistant bacteria. So unfortunately, farmers have used, with their livestock, added antibiotics to animal feed. Doctors may have overprescribed antibiotics. You go in, you're sick, and you want to leave with antibiotics, but you could have a virus. And so by overprescribing antibiotics, that has also led to antibiotic-resistant bacteria. And then people may not take their full course of antibiotics, allowing mutant bacteria that may be killed more slowly by the drug to survive and multiply. So you're supposed to take antibiotics for 10 days, and let's say day 4, you feel so much better and you stop taking it. That is what's going to cause antibiotic-resistant bacteria. So natural selection for antibiotic resistance is particularly strong in hospitals where antibiotics use is extensive. In fact, there is a formidable drug called a superbug known as MRSA, which is methicillin-resistant Staphylococcus aureus, and can cause potentially fatal, that means will kill you, systemic whole body infection, and often begins in community settings such as athletic facilities, gyms, schools, military barracks. It is absolutely everywhere. But in normal circumstances, your immune system will fight it off. It is where you have individuals with compromised immune system that really, if they do get MRSA, unfortunately, can be fatal. All right, and with that, we are finished with chapter 13. Move on. Okay. Now, we're going to continue our talk with evolution and look at it on a species level. Okay, so we're going very specific. And so we're going to dive into how the biological diversity evolves. So let's talk about human and our footprint. So humanity has extraordinary effect on the ecology and geology of Earth. Our indelible footprint includes the transport of organisms from their natural homes, the prevalence of agriculture and domesticated animals, the existence of manufacturing materials such as plastic and concrete, radioactivity from testing nuclear weapons, and climate-altering emissions from burning fossil fuels. So the irreversibility of these changes has led to scientists to propose that a new epoch in Earth's history has begun, the Anthropocene. So the Anthropocene signals a significant shift in the geological record that includes a high rate of extinction and accelerating change to Earth. So human-driven changes in the environment also bring about evolutionary change in populations of organisms, like we just said in chapter 13, which include the pesticide-resistant insects and antibiotic-resistant bacteria. All right, so just as a quick recap, we talked about Darwin and his book on the origin of species by means of natural selection. Well, the diversity of life evolved through speciation, which is the process in which one species splits into two or more species. So species is a Latin word meaning kind or appearance. So the biological species concept defines a species as a group of populations whose members have the potential to interbreed with one another in nature and produce a fertile offspring. And fertile offspring is very important because that means that offspring can also reproduce. So the biological species concept cannot be applied in all situations, including asexual organisms and fossils. But here you can see the biological species concept is based on reproductive compatibility rather than physical similarities. So you have a similarity between different species. And then, of course, you see here there's diversity within one species. So what is a species? Well, biologists may also define a species according to its measurable physical traits, solely on the basis of molecular data or sort of barcode that identifies each species, or as the smallest group of individuals sharing a common ancestor and forming one branch on the tree of life. So there reproductive barriers between species is anything that prevents an individual of a closely related species from interbreeding. And so there's two types of barriers: prezygotic barrier, which prevents mating or fertilization between species, and then postzygotic barrier, which operate if interspecies mating occurs, the hybrid zygote forms. So here is a picture of the iguana. You have the marine iguana on the right, remember, with the flat tail that aids for swimming. And then you have the land-dwelling iguana, and they're both unique to their specific environments or their habitat in the Galapagos Islands. Examples of prezygotic barriers. An example is temporal isolation, where you have these skunk species that mate at different times, so they'll never mate. You have habitat isolation, as we just talked about. You have garden snake species, but they have different habitats, one in land versus one in the marine. You have behavioral isolation, where you have the mating ritual of the blue-footed booby, and the female will choose her mate. You have mechanical isolation, where the hummingbird is compatible with one flower versus on the left versus the other flower. And you have gametic isolation, where the sea urchin species whose gametes cannot be fused. Now, examples of post-zygotic barriers. Remember, if mating does occur, will produce a reduced hybrid viability. It also will produce a reduced hybrid fertility. So in this case, you have a horse and a donkey that's made, the mule is infertile. And then you can also have a hybrid breakdown. So the one I want you to remember is an example of a post-zygotic barrier is reduced hybrid viability. So this is a fragile salamander. Now, mechanisms of speciation. A key event in the origin of a species occurs when a population is somehow cut off from other populations of the parent species. So species can be formed by allopatric speciation or sympatric speciation. So with allopatric speciation, it is the initial block of the gene flow is a geographic barrier that physically isolates the splinter population, or sympatric speciation is a block, but it's without geographic isolation. So a variety of geological process can isolate populations. Speciation can occur with the evolution of reproductive barriers between the isolated populations and its parent. And even if two populations should come in contact with each other later on, that reproductive barrier will keep them as separate species. So here you can see an example of an allopatric speciation with the antelope squirrel on opposite rims of the Grand Canyon. So with sympatric speciation, a new species arises within the same geographic area as its parent species. So factors that can reduce gene flow in sympatric populations include polyploidy, in which a species may originate from an accident during cell division that results in an extra set of chromosomes, habitat complexity, and sexual selection. So many plant species we grow for food are polyploids, including oats, potatoes, bananas, strawberries, peanuts, apples, sugarcane, and wheat. So we saw, or in the island showcase of speciation, the Galapagos Islands are one of the world's great showcases of speciation. For example, the island has 14 species of closely related finch. So these birds share many finch-like traits, but they differ in their feeding habitats. Their habitat, feeding habits, their habitats, and their beaks, which are specialized for what they eat. The distinctive beaks adapted for the specific diet of the different species of finch are examples of the correlation between structure and function. So as you can see here, based on what the finch eats, their beaks are adapted for that specific diet. Okay, going back to fossil record, we know that fossils are evidence of organisms that lived in the past. And the fossil record is the sequence in which fossils appear in the rock strata and an archive of macroevolution. The geologic timescale divides Earth's history into a consistent sequence of geological periods. And right now, we are in the Cenozoic period. So that's very important for you to remember. We're in the Cenozoic period. So with the fossil record, the most common method geologists use to learn the age of rocks and the fossils they contain is radiometric dating. And this is a method based on the decay of radioactive isotopes. Another commonly used method to date layers of volcanic rock or ash above and below the sedimentary layer in which the fossils are found. And by inference, the age of the fossil is between those two dates. So mass extinctions have occurred and are explosive diversifications of life. The fossil record reveals there have been 5 mass extinctions over the last 540 million years. And in each of these events, 50% or more of Earth's species have died out. So extinctions seem to have provided the surviving organism with new environmental opportunities. So you have the Permian mass extinction that claimed about 96% of marine species. And then you have the Cretaceous extinction that occurred about 66 million years ago and included the extinction of all dinosaurs except for birds. So with mass extinctions were followed by a period of evolutionary change, which means that new species arose as survivors and they became adapted to occupy new habitats or to fill community roles vacated by extinction. So the first utility of feathers may have been for insulation, but once flight became an advantage, natural selection would have gradually remodeled feathers and wings to fit their additional function. So structures such as feathers that evolve in one context, but became co-opted for another function are called exaptations. Now, let's talk about classification. So we already talked about taxonomy, and that is the naming and classification of species. Well, systemics includes taxonomy and focuses on classifying organisms and determining their evolutionary relationships. So with that, biologists use phylogenetic trees to depict the hypothesis of the evolutionary history of species and reflect their hierarchical classification of groups. So here's an example of the relationship of classification and phylogeny for some members of the order Carnivora. And again, you read this from left to right. Now, identifying homologous characters. So homologous structures in different species may vary in the form and function, but exhibit fundamental similarities because they evolved from the same structure in a common ancestor. So homologous structures are one of the best sources of information for phylogenetic relationships. Now, convergent evolution involves superficially similar structures from different evolutionary branches that result from natural selection shaping analogous adaptations. So similarly, due to convergence is called analogy and not homology. So you can compare the embryonic development of two species, and that can often reveal homology that is not apparent in the mature structures. And if homology reflects a common ancestry, then comparing the DNA sequences of an organism gets to the heart of the evolutionary relationship, meaning that the more recently two species have branched from a common ancestor, of course, the more similar their DNA sequence should be. And then some fossils are preserved in such a way that DNA fragments can now be extracted and compared with living organisms. So when we want to infer phylogeny from homologous characters, we look at cladistics, which is organisms that are grouped by common ancestry. And a clade consists of ancestral species and all its evolutionary descendants and forms a distinct branch in the tree of life. Thus, identifying clades make it possible to construct classification schemes that reflect the branching patterns of evolution. So cladistics is based on Darwinian concept of with modification from a common ancestor. So to identify clades, scientists compare an in-group with an out-group. And an in-group is the group of species that actually is being analyzed, and the out-group is the species or group of species known to have diverged before the lineage that contained the group being studied. So phylogenetic trees are hypotheses about evolutionary history. And Linnaeus divided all known forms of life between the plant and animal kingdoms. So he basically had a two-kingdom system, and it prevailed in biology for over 200 years. Then in the mid-1900s, the two-kingdom system was replaced by a five-kingdom system that placed all prokaryotes in one kingdom and divided eukaryotes among four kingdoms. Then finally, in the late 20th century, molecular studies and cladistic led to the development of what we currently have, which is the three-domain system that consists of two domains of prokaryotes, bacteria and archaea, and then one domain of eukaryote. So the domain of eukaryote is currently divided into kingdoms, but the exact number of kingdoms is still under debate. So what you have now is our three-domain classification system that we're currently using. And then of course you see with eukarya, it's broken down further into those kingdoms listed. Okay. And with that, we are finished with chapter 14. All right, so real quick, I want to just kind of do a quick recap of where we are and next steps. Okay, so today we covered chapters, we're almost finished, chapters 13 and 14. So Wednesday, you have your vocabulary 13. Monday, you have your vocabulary 14 quiz, and then we will circle back for our last Zoom on Wednesday, April 10th. So that will be our last chapter on ecology before your final Unit 5 exam, okay? All right, again, thank you so much for showing up. And I also want to say thank you so much for your efforts in the class. You all are doing tremendously, and I really appreciate all your efforts and continue the good work. We just got a little bit more, so continue to push through and do your very, very best. Have a wonderful evening.
Thank you, Professor.
Thank you, Professor. Good night.